such as enzymatic fuel cells, microbial fuel cells, and biophotovoltaics. And it will be followed by the role of nanomaterials in the biological systems. And finally, I will briefly explain the work carried out at CSIR SECRI with regard to the biofuel cells. Energy, as you all know, energy is the hot currency which is in strong demand at the global level. It is an important factor in the advancement and development of human society. There are different forms of energy such as heat, light, mechanical energy, electrical energy, and so on, which are interconvertible in nature. But mostly our energy demand is supplied by fossil fuel technologies, which will not last forever. Also, it results in lot of problems related to pollution and global warming. As you might all be aware of the science documentary which was released in the year 2006, An Inconvenient Truth, which was presented by the then American Vice President Al Gore in 2006, which fetched Nobel Prize for him and the panel on climate change in 2007. This film is about raising international public awareness of global warming and re-energizing the environmental movement. It is also about Al Gore's campaign to educate people with regard to global warming. Yeah.
I'm sorry to interrupt, ma'am, but your PPT is not visible, ma'am.
Not visible.
We couldn't see your presentation, ma'am.
Okay. 1 second, please.
Yes, ma'am.
Ma'am, in the bottom right corner, there is a button now button. Bottom right corner. You please move the mouse, then you can see.
Right corner.
Bottom right. Present now. There is a button. Present.
Yeah.
You click on that.
You are okay.
Then there is a window. There are 3 options. You click on a window.
Window, okay.
Now inside you can see your PowerPoint.
Yeah.
You click on the PowerPoint, then immediately there is a sharing option. It will come.
Okay.
You just click on this.
Now is it okay?
Yes.
Is it visible now?
Yes. Yes.
Yes, ma'am.
Okay. Should I start from the start?
No. You can continue, madam. You don't have to go first.
Okay. Okay. This documentary I was mentioning about is called by the name An Inconvenient Truth. This science documentary fetched a Nobel Prize for the then American Vice President Al Gore in 2006. It is about his campaign to educate people about global warming. Also, this film raised awareness, international public awareness of global warming and re-energizing the environmental movement. This is all necessary because we are consuming the fossil fuel reserve at a faster rate. Also, the fossil fuel based technologies release lot of polluting gases, carbon emissions, and so on, which result in climate change. So, it results in global warming. Also, if you see the world energy demand, you can see the major fraction of the energy demand is met by fossil fuel cells and fossil fuels, and only a small fraction is met by non-fossil fuels, which are nuclear, hydro, and biomass. And you can see that fraction that is met by wind and solar energy is so low. And also, if you compare the energy demand with the population, the energy demand is expected to increase at a rate of 63 to 160%, compared to increase in population expected is 6 by 60%. And also, if you see the consumption of different forms of energies, you can see the major energy demand is fossil fuel based. Petroleum, natural gas, coal constitutes 24%, 23%, and 39% respectively. And when you come to renewable energy, biomass occupies 47% of the energy consumption. So people are in, since the fossil fuel based technologies results in global warming and all sorts of pollution problems, there is always a substantial growing demand for alternative energy conversion technologies and for alternate renewable energy sources. So in that connection, electrochemical energy source has a substantial growing demand for advanced electrochemical energy systems such as high energy batteries for transport vehicles, stationary energy storage, next generation fuel cells with high efficiency, membrane reactors, and so on. And you can see the electrochemical conversion and storage and its role in hydrogen economy in this slide. So the hydrogen economy is going to play a big role in the coming years. So the hydrogen is liberated by the electrochemical water splitting process, which can be used in fuel cells for the supply of electricity, and it can be hydrogen can be burnt as a gas, and can be used in hydrogen vehicles. It can be combined with nitrogen to form ammonia and so on. So this hydrogen can be electrochemically converted into electricity by fuel cells. Other electrochemical storage devices are capacitors, supercapacitors, batteries, and hybrid systems. Coming to the biological systems, there are 3 different ways by which a biomass can be converted into energy. Mostly from the biomass, 2 different types of gases can be obtained as fuels. One is hydrogen, the other one is methane. Mostly biomass is being utilized at different levels for heating purposes, for small scale heating purposes in households. Medium scale users typically burn wood chips in boilers. Large scale boilers are used at the industrial level. For bioelectricity applications, conversion of energy, chemical energy available in biomass to electrical energy is possible in 3 different ways. One is microbial fuel cells, the other one is enzymatic fuel cells, the third one is biophotovoltaics. So these are the 3 main conversions technologies that will be available in future for electrochemical energy conversion from biological systems. You may wonder how electrochemistry and biology are related. Electrochemical reactions and biological electron transfer reactions, both are charge transfer reaction happening under the influence of electrochemical potential gradient. Both are heterogeneous reactions, both are pH dependent, and biological cells and electrochemical cells exchange mass and energy across membrane by electrochemical processes. Also, you may all know about the signal transmission in nerve cells is purely electrochemical in nature. It depends upon the potential dependent conductance changes of K plus and Na plus ions across the nerve cells that is responsible for signal transmission in nerve cells. So Always a rest potential of 80 to 100 millivolts is seen in the biological membrane potential. Also, I can tell you another example. You might have all known about electric eel, which is an electric fish. They are also called as biological batteries. They have electrocytes, which are electric organs arranged in long stacks. These fish some of these fish can even liberate an electrical voltage of 600 volts. Mostly these electric fish produce this voltage for echolocation of the spray. So these two examples tells us that electrical energy can be easily obtained from biological system. So over to the definition of fuel cells. Fuel cells are environmentally friendly electrochemical devices to directly convert chemical energy into electrical energy without intermediate steps. Mostly in conventional fuel cells, we use noble metals such as platinum, ruthenium, palladium, or their alloys as catalyst for the oxidation of pure fuels at the anode and the reduction of oxygen at the cathode. This is a electrochemical fuel cell. Now, how we can obtain energy conversion similar to a fuel cell device from biological system. So there are many sources of energy within the human body. The human body can release heat energy. It can release chemical energy of organic molecules such as glucose. It can also liberate mechanical energy in the form of such as breathing and motion of limbs. So energy harvesting devices that can convert these type of physical and chemical body energies to electrical energy can be developed using biofuel cells. For example, in this figure, you can see body heat can give rise to 2 to 5 watt. Biochemical fuels can give rise to 100 watts, and arm motion can give rise to 60 watts. If you see the energy consumption of a pacemaker, implantable devices like pacemaker requires a power of 10 microwatts to 30 watts. Insulin pump requires a power of 70 microwatts. Cochlear implant requires a power of 0.02 watt to 1 watt. So there is a lot of scope or lot of possibility for developing enzymatic fuel cells for biomedical applications such as hearing devices, neurostimulators, insulin pumps, incontinence devices, bone growth stimulators, pacemakers, artificial hearts, drug pumps, and so on. So the main goal of these researchers is to find out, develop a implantable fuel cell in the millivolts to watt range. So coming to the definition of enzymatic fuel cell, it consists of a 2 electrode cell. It is exactly similar to that of a electrochemical fuel cell. It is separated by a proton conducting medium, which can also act as an electrolyte, and 2 appropriate enzymes are used as catalyst at the anode and at the cathode. And the cathode, mostly oxygen or peroxides are reduced to water. The advantages of this type of fuel cells is that the catalyst is renewable. Redox enzyme can be extracted from a wide range of living organisms in a renewable manner. The fuels can be diverse. For example, fuels such as sugars, alcohols, organic acids, hydrogen, and mixture of these materials can be digested by living organisms. The operational conditions are very mild and safe. Also, the redox enzyme provide exceptional specificity towards their substrates, therefore allowing the assembly of bioanode and biocathode in a membraneless cell, so that miniaturization of these enzymatic fuel cells are possible. So usually for anodic reactions, the enzymes that are used are dehydrogenases and oxidoreductases. And for cathodic reactions, enzymes such as cytochromes, peroxidases, bilirubin oxidase, and lacase are used as catalyst. So this slide shows the evolution of enzymatic fuel cells over a period of, say, starting from 1970 to 2010. So initially, the enzymatic fuel cells were inserted in animals, and the possibility of development of enzymatic fuel cell was tested. Later on, it was tested on fruits like grapes, and later on using some sea animals. And nowadays, researchers are trying to make implantable devices that can be used in the human body. This example shows the example of an enzymatic fuel cell device, which is within few nanometers, say around within 20 nanometers size biofuel cell made up of enzymes. So in 2012, in Case Western Reserve University, there was a report on an implantable biofuel cell in a live insect. So this is a cockroach in which were inserted needle-like electrodes, where two enzymes were used on the anodic side, one which converts triglyceride sugar to glucose, and the second enzyme was glucose oxidase, which converts glucose to gluconic acid. On the cathodic side, bilirubin oxidase was used, which reduces oxygen to water. So from this fuel cell, some microwatts of energy could be produced. And similarly, in Clarkson University, another implantable fuel cell was developed based on CNT immobilized enzymes. Even the fuel cell was implanted on a snail, and this university was also able to show electrical energy could be generated in the implanted fuel cell. But there are several challenges associated with the enzymatic fuel cells. For example, the inability to complete oxidize fuels, low power density, poor operational stability, limited voltage output. So these are the challenges to be faced when one is trying to develop a enzymatic fuel cells. Also, when you use one or two enzymes, it becomes impossible for the complete oxidation of the fuel. Therefore, the power output decreases to a large extent. For example, in an ethanol fuel cell, based on 20% volume by volume ethanol, incomplete oxidation leads to a decrease from 5,442 to 362 watt-hour per liter. So such a huge difference in the power output will be observed if the oxidation is incomplete. So the main issue with all these enzymes is the electrical connection to the enzymatic redox center to the electrode should be highly efficient. So in that connection, nanotechnology offers exciting tools to achieve efficient electrical wiring or interfacing between redox enzymes and electrical circuitry while providing highly active surfaces. So I will list out from the literature what are all the nanomaterials that are used in enzymatic fuel cells to improve electron transfer and improve energy conversion efficiency. This will be followed by my work at the later point. So carbon nanotubes are largely used in enzymatic oxidations and enzymatic fuel cells because they have a nanowire morphology and they are found to be biocompatible and they have excellent conductivity, high 3D electroactive area, and it increases surface concentration of enzymes and other redox molecules. Therefore, one can achieve direct electron transfer in the presence of carbon nanotubes. So this slide also explains what are the different methods by which an enzyme or a mediator can be attached to carbon nanotube. So either we can attach by electrografting or by cycloaddition or by supramolecular pi stacking or by introducing redox polymer composites or by using covalent linkage using amide coupling. So there are different methods by which a redox mediator or an enzyme can be coupled to the carbon nanotubes. So carbon nanotubes have to be functionalized with redox molecules, and these are the methods that are used to functionalize them. Another method is to incorporate the carbon nanotubes along with the polymer complexes. So mostly in literature, if you see, polypyridyl osmium complexes bound to a polyvinyl pyridine polymer backbone is being used along with CNTs. So to this backbone, glucose oxidase and bilirubin oxidase will be immobilized on the polymer chain, and to this matrix, dispersion of CNT in SDS, sodium decyl sulfate, will be introduced. So in this configuration, a fuel cell exhibiting 740 microwatt per centimeter square at 0.57 volt has been reported with bilirubin oxidase. BOD means bilirubin oxidase at the cathode and glucose oxidase at the anode. Also, other carbon nanomaterials such as graphene and mesoporous carbon are being used for direct electron transfer. Other type of nanomaterials like clay and metal nanoparticles and metal oxide nanoparticles are also being used in enzymatic fuel cells to improve the electron transfer efficiency. And there is a report about the first example of direct electron transfer enzymatic fuel cell in the presence of multi-walled carbon nanotube composite. The fuel cell that was developed was a mediatorless glucose oxygen fuel cell. The direct electron transfer was observed at the anode compartment, which is glucose oxidase CNT anode. Also in the cathode compartment, lactase CNT cathode. So in this fuel cell, high power output of 1 milliwatt per centimeter square could be obtained at an OCV, open circuit voltage, of 0.95 volt. And another important feature is that this material was found to be highly porous and highly conductive. So it favors the diffusion of substrates to the enzymes, and the electron mobility was improved to a large extent. So this figure shows the power curve that is recorded for the direct electron transfer fuel cell, and this is the discharge curve, and you can see the size of the enzymatic fuel cell that is developed, that is shown along with the scale here. So other type of nanoparticles that are being used in enzymatic fuel cells are clay nanoparticles, also layered double hydroxide. This example shows the layered double hydroxide can immobilize both CNT as well as the enzyme molecules, so that direct electron transfer can be observed. Here also in the cathode and the anode, both the layered double hydroxide are used along with the mediator ABTS. ABTS is 2,2-azino-bis 3-ethyl benzathiazolene 6-sulfonate. This is a mediator, and this is used along with the lactase to produce direct electron transfer, and the maximum current density observed here in this case is 77.6, which is higher than 28 microamperes, which is observed without CNTs. Also, many other examples like gold nanoparticles along with polyaniline are being used at the glucose dehydrogenase at the bioanode and bilirubin oxidase at the biocathode. the biofuel cell delivered 32 microwatts per centimeter square power output and 0.5 volt OCV. Also, another example I can tell you here is gold nanoparticle sol gel matrix, which delivered a power output of 1.56 milliwatt per centimeter square at 550 millivolts. Also, many polymers, redox polymers like polypyrrole, P. Polyvinylpyridine are being used in the biofuel cells. In this example, glucose oxygen fuel cell has been integrated in a fluidic system on a hydroquinone sulfonate glucose oxidase anode, where polypyrrole electrodes were designed by electropolymerization within the pores of anodized aluminum oxide. One more example of the application of polymer is given here. Here, nano biofuel cell made of single naphthalene polyvinyl pyrrolidine nanowire connected to lac-S cathode and a GOX CNT anode delivered 0.8 to 0.3 microwatt. Here they have made a single nanowire made up of naphthalene polyvinyl pyridine, which connected the cathode and the anode, and this could deliver a current power output of 0.8 to 3 microwatt. So the next biological system that can be used for the production of electrical energy is microbial fuel cells. Microbial fuel cells are bioelectrical devices that harness the natural metabolism of microbes to produce electric power directly from organic material. The performance of microbial fuel cells depends upon the factors like anode material, cathode material, the substrate which is used as a catalyst, polymer electrolyte membrane, and so on. So the cell voltage that is obtained is subjected to losses, some of the losses with respect to the electrodes and the diffusion of the substrate to the electrode surfaces. There are also many other factors which limit the MFC technology. Micro MFC is microbial fuel cell technology. We always rely on expensive catalyst and prohibitive amounts of catalyst, especially platinum, and ineffective use of catalyst. For example, poor dispersal of the catalyst, and also reliance on expensive reagents, materials for optimum function, especially the addition of electron transfer mediators and electrode materials. Further, the toxicity of electron transfer mediators, low power generation, poor durability of electrode materials, poor scalability, poor mass transport across the system and within compartments, and the requirement for external power input for optimum function, example agitation and heat. Also, there are several other factors which contribute to the low power density that is observed in microbial fuel cell. The first one is surface area and porosity. The power generated by microbial fuel cells is greatly influenced by the surface area of the electrodes. Increasing the surface area to volume ratio enhances the efficiency of the fuel cell. Increasing the surface area leads to increase in the number of reaction sites and increases the movement of electrons. However, greater porosity also leads to lower electrical conductivity. The second factor is electrical conductivity. Using an electrode material with high electrical conductivity, the movement of electrons from the anode to the cathode through the electrical circuit becomes easier, which leads to an increase in throughput of the fuel cells. Stability and durability. Due to oxidation and reduction reactions occurring at the anode and cathode, there is decomposition and corrosion of the electrode material. Thus, to avoid unnecessary cost increases without affecting the efficiency of the fuel cell, electrode material should be durable and stable in both alkaline and acidic environments. The last aspect is cost and accessibility. The cost of a material is crucial in the development of a fuel cell. For commercial fuel cell application, component materials should be low cost and accessible. For instance, platinum is widely used electrode material, but it is extremely expensive and non-sustainable. Therefore, composites of precious metals such as platinum are generally better alternatives to the metal alone. So these are generally the limitations that we come across when we try to develop a microbial fuel cell. The conventional electrode materials that are often used in microbial fuel cells are graphite flakes, carbon paper, carbon cloth, carbon brush, carbon rods, and metallic materials like stainless steels, copper sheets, apart from platinum. Now, the decoration of these electrode materials with nanoscale materials efficiently enables extracellular electron transfer process from microbes to anode surface. So when we incorporate nanoscale materials along with these electrode materials, the electron transfer rate increases to a large extent. The optimal nanostructured material is both rich in micropores and nanopores, in which the microporous structure is conducive to the growth of biofilm, and the nanoporous structure can enhance the direct electrochemistry of electronic medium for further achieving efficient biocatalysis and electrocatalysis. So carbon nanomaterials are mostly being used in microbial fuel cells. Again, carbon nanotubes play a significant role in the development of microbial fuel cell. It is of tremendous interest, as I have told you, in the case of, enzymatic fuel cells. Its special fiber structure, high mechanical strength, toughness, large surface area, good thermal stability, chemical inertia, strong conductivity, and unique one-dimensional nanometer scale material all contribute to the significance of carbon nanotubes in the development of microbial fuel cells. So carbon nanotubes modified anode leads to high power densities than the unmodified anode because it increased the number of electrochemically active sites induced by complicated networks and enhanced attachment of microbial film. So there is a report on the in the literature about modified carbon paper with multi-walled carbon nanotubes as the anode through layer by layer self-assembly method compared with plain carbon paper. The modified carbon paper with CNTs in MFC had a low internal resistance of 258 ohms, which was significantly lower than the MFC operated with carbon paper alone. So with carbon paper alone, the internal resistance was 1163 ohms. In presence of CNTs, it decreased to 258 ohms. Meanwhile, the highest power density reached to 290 millivolts per meter square from initial 241 millivolts per meter square. So there are other reports in the literature about the use of carbon nanotubes. One of the groups have developed carbon nanotubes with 3 different lengths, 8 micrometer, 13 micrometer, and 19 micrometer. A low modification density with a length of 19 micrometer achieved maximum power density of 3,360 milliwatt per meter square, which was 7.4 times higher than the carbon cloth electrode. The longer CNTs provided more active sites for electron transfer of bacteria, and the space between CNTs was large enough for the bacteria's proliferation, resulting in higher power output. In another report, a novel combination of hydrothermal liquefaction and CNTs fixed bed MFC for corn straw biomass treatment was discussed. CNT is coated on carbon fiber felt as the electron acceptor. The fixed bed MFC obtained the maximum power density of 680 milliwatt per meter cube at the organic load of 2.41 gram liter per day, and the high efficiency MFC reactor could be used repeatedly, which extended the service life and increased the stability of electricity generation. Also, some of the researchers reported about cytotoxicity of CNTs. So mostly the amount of CNTs that should be added to the electrode material should be only about 20 to 30% of this 30% of the total catalyst composition. Otherwise, some of the researchers have observed the CNTs to be cytotoxic in nature. The other carbon nanomaterial that is often being used in microbial fuel cell is carbon nanospheres. The organic matters can be transferred to solid phase to form uniform sizes, better appearance, and higher specific surface area by high temperature carbonization and activation. So by subjecting organic material like glucose to controlled carbonization and activation, we can get beautiful carbon nanospheres which exhibit excellent biocompatibility and electrical conductivity in long-term operations. Also, it is observed the space among the nanospheres was abundant and the nanospheres were interconnected. The particular structure enhanced the bacteria adhesion on this interface, and the electrochemical activity improved the extracellular electron transfer process. Additionally, the decorated graphite with carbon nanospheres largely improve the power density. The prepared carbon nanospheres also exhibit long-term durability and non-toxicity to the environment. So it is it has high potential for practical application for preparing a stable anode. The another frequently used nanomaterial is graphene. Graphene, as you all know, is a two-dimensional periodic honeycomb lattice of carbon 6-membered ring. So it can warp into zero-dimensional fullerene or roll into one-dimensional carbon nanotube or stack into three-dimensional graphite. So it is a basic unit of other graphite materials. Compared with conventional carbon materials, graphene shows excellent electronic conductivity, high specific surface area, strong strength, good stability, and biocompatibility. So one of the reports where they have used graphene to modify stainless steel mesh reported a maximum power density of 2,668 millivolts per meter square, which was 18 times higher than stainless steel mesh as anode in MFC. Graphene was favorable for enriching bacteria, which facilitated electron transfer from bacteria to anode. In another report by Yu, they have constructed MFC with graphene aerogel as the bioanode by a hydrothermal reduction method. The maximum power density reached up to 5 watt per meter cube, which was higher than the carbon cloth as anode. The improved power output was attributed to the enhanced electronic conductivity and anode surface area. The other novel electrode material that is being evaluated nowadays is a diamond film. As a new type of electrode, diamond materials exhibit the superior application prospect in electrochemical sensor and electrocatalysis. The diamond itself shows high mechanical strength and more environmentally friendly. Nanometer amorphous diamond film has no toxicity to human health and good biocompatibility. So one of the reports, they have prepared diamond nanoglass electrode by using ion etching and oxygen plasma process on concentrated diamond film with the microorganism Schwannella lohickica as the electrogenic bacteria. The MFC delivered a stable current density of 1.2 microamperes per centimeter square, which was 3 times higher than the control group. So this may benefit from the nanograph morphology in effective enhancing of the electron transfer ability. So another popular material that is being used as electrode material is activated carbon. Activated carbon with porous structures are often used for organic adsorption, which are originated from charcoal, various fruit shells, and high quality coal as raw materials. Unlike graphene and diamond nanoscale materials, the fabrication of cost activated carbon is relatively low, and the large accessible surface area is beneficial to improve anodic performance. For example, chestnut cell, bacterial cellulose, polyacrylonitrile fibers are being used to produce activated carbon. So apart from carbon-based materials, metal-based nanomaterials are also being used in the microbial fuel cells. Metal-based materials has a much higher conductivity than carbon-based materials. However, they are not widely applicable by contrast with carbon materials as they are easily corroded. Currently, nanostructured metal materials with high catalytic activity, conductivity, and biocompatibility have attracted their utilization of anode decoration in microbial fuel cells. Then the next type of material is polymer-based nanomaterials. Conductive polymer with high electronic conductivity and stability have drawn much attention among researchers to explore the potential as nanostructured materials to enhance energy generation of microbial fuel cells. Polyaniline carbon nanotube composites, carbon cloth P dot, carbonized cotton textile modified with nanostructured polydopamine are some of the polymers that have been employed as electrode materials in microbial fuel cells. So this table, it lists out, it gives you an example of different varieties of nanoparticles that are being used in the microbial fuel cells for current generation. So this table lists out different type of materials like AU nanoparticles, Mn4 plus graphite, graphene nickel composite electrode, Fe3O4 CNTs, carbon felt, SnO2 RGO carbon felt, RuO2 carbon felt, and so on. And this column tells you the different kinds of biocatalyst, the bacteria that are used in the microbial fuel cell. The performance of the corresponding fuel cell is listed out here. So you can see energy as output, the power output as large as 3903 milliwatt per meter square can be obtained in the case of the microbial fuel cells developed with these nanomaterials. So the next type of biological system which can produce electrical energy is biophotovoltaics. So it is photoelectrochemical energy conversion in biological systems. So the photosynthesis process, we are trying to insert the catalyst into this photosynthesis process, which is again water splitting, and we try to tap the energy of photosynthesis as electrical energy. So metal and metal oxide nanoparticles can be incorporated both internally and externally with biological organelles and cells to increase light harvesting efficiency and improve electron transport to electrodes. While light absorption plays an important role in improving light harvesting conductivity is key to effective electron transport. Nanoparticles are used for this, and mostly they are found to be biocompatible. So next I will explain about the work carried out by me at CSIR Secre, which is related to enzymes and microorganisms. So with respect to the enzymes, we have to work on the anodic part separately and the cathodic part separately. As we were working on the sensing system as well, we always try to find application as a sensor material and also try to find out a material that can be used as an anode in the enzymatic fuel cells. So we have developed different kinds of strategies to link enzymes to the electrode surface so that we hope to attain maximum electron transport efficiency. In my earlier slides, when I explained about enzymatic fuel cell, I was trying to explain that the main issue in the development of enzymatic fuel cell is the linking the prosthetic group of the enzyme, which is responsible for electron transfer, to the electrode material efficiently, or the electrical wiring of enzyme to the electrode surface should be efficient. Then only we can have a very good electrode system for a sensor as well as for fuel cell. So we have adopted different kinds of strategies to immobilize the enzyme on the electrode surface. The first strategy that we have tried is self-assembled monolayers. Self-assembled monolayers are now monomolecular films of thiol molecules on gold electrode surfaces, which are formed without a help of any external force. So they self-assemble on gold electrodes in the form of a monolayer, and we try to make use of thiol molecules with suitable terminal groups like amino groups, carboxylic acid groups, and so on, so that to these terminal groups we can attach either a mediator or an enzyme to the electrode surface. So this slide talks about the incorporation of a mediator and glucose oxidase to cysteamine monolayer. Cysteamine monolayer, it is ethane thiol. So to the cysteamine monolayer, we have incorporated the 1,4-diamino anthraquinone. Diamino anthraquinone is attached to this, which is a mediator covalently to this thiol molecule, which is then covalently linked by glutaraldehyde linkage by glucose oxidase. So on this side, you can see the example of a mediator, diamino anthraquinone, and two different enzymes. One is invertase, other is glucose oxidase. So this unit can be used for sensing sucrose, and this unit can be used for sensing glucose. So here glucose oxidation occurs, here sugar oxidation can occur. The potential of oxidation is sufficient enough, we can use this in the enzymatic fuel cell. Also, in the literature, so many applications I have discussed with the help of carbon nanotubes, graphene and other carbon-based nanomaterial, but we have used a new type of molecule, a dendrimer molecule, for attaching enzymes to our electrode surface. So the dendrimers or molecules have special features. They have a core-like structure, they have branching units, they have internal cavities, they have closely packed surface groups. So it has a globular structure. So this type of dendrimer molecules have different generation, 1, 2, 3, 4, like that. Different generations of dendrimer are available in the market. We have made use of PAMAM dendrimers for our research purpose. Polyamidoamine dendrimers of 4th generation and 5th generation are used in our work. We have chosen this molecule because in the internal cavities, nanoparticles can be encapsulated. So instead of using a single molecule, this a single dendrimer molecule can encapsulate a number of nanoparticles. Therefore, we get increase in the number of active sites, therefore increase in the electron transfer rate. Also, you can see on the peripheral groups, there are numerous groups in a single molecule. For example, a fourth generation dendrimer will have 64 functional groups on the outside. So you can attach 64 nanoparticles. Suppose if we can link efficiently 64 nanoparticles to a single molecule. So again, it effectively increases the number of active sites. Therefore, you can always expect to get an increase in surface area. So making use of this polyamidoamine amine terminated dendrimer molecules, we encapsulated gold nanoparticles inside the dendrimer molecule. Then we attached the glucose oxidase enzyme, which can be converted to glucose on oxidation. In presence of gluconic acid, it is converted into gluconic acid. Initially, we tried as a biosensor. So making use of this AU dendrimer nanocomposite, we can sense glucose in the range 1 to 10 millimolar. We also found the number of layers of these dendrimers could be increased. So we formed 3 layers. Initially, we tried with 1 layer of dendrimer molecule. Then we formed 3 layers of molecule, and we could increase the range of detection up to 25 millimolar. So by increasing the number of layers, we can increase the range of detection. So in another work reported by us, we electrografted these dendrimer molecules on graphene surface. So it is an the dendrimer molecules can aminate the graphene surface. So the graphene modified GC electrode was electrografted to the dendrimer molecules, to which these are the SEM image of the graphene grafted dendrimer molecules. And to this molecule, peroxidase enzyme was initially grafted. So it was used for analysis of hydrogen peroxide in blood serum. Later on, we extended this method for the detection of glucose. So by enzymes, 2 enzymes are attached to this graphene-based electrode. So initially, graphene was modified on glassy carbon electrode, to which the dendrimer molecules were electrografted. Then to the electrografted dendrimer molecules, horseradish peroxidase enzyme was attached. Then the glucose oxidase enzyme was attached, so which was used for sensing glucose. So bioenzymatic glucose sensor. Also, we have attached cholesterol oxidase enzyme to glassy carbon electrodes using trimetallic nanocomposite. We have used copper, platinum, bismuth nanocomposite for the immobilization of cholesterol oxidase. And we have shown the cholesterol on oxidation gives to cholesterol plus H2O2. The obtained H2O2 can either be oxidized or reduced. So we are able to do the oxidation of hydrogen peroxide as well as reduction of hydrogen peroxide. So we can sense the cholesterol either anodically or cathodically. So that we have achieved in this work. Also, this shows that this cholesterol-based electrode can either act as an anode or act as a cathode if it is used in a enzymatic fuel cell. So this table tells you that how our electrode is found superior compared to the other reported work in the literature. Also, we have used different type of compounds metal organic frameworks we have used for the immobilization of enzymes. We prepared bismuth-based metal organic compound using the ligand 2,2-dithiodibenzoic acid, and we have incorporated glucose oxidase enzyme onto this metal organic framework, and we have observed the direct electron transfer of the enzyme. That is, we can directly wire the enzyme to the electrode surface. So we can see the FAD electrochemical signature. FAD is the prosthetic group of the glucose oxidase enzyme. The electrochemical signature of the FAD molecule is seen in the cyclic voltammogram. The surface coverage of the enzyme molecule is found to be of the order of 10 power minus 12 mole per centimeter square. And you can see how the glucose oxidation takes place in the oxygen-free solution. So in the oxygen-free solution, glucose can be oxidized. That shows the glucose oxidase enzyme is electrically wired. Oxidation occurs only through the enzyme, and no other mediator comes into role here. So we were able to demonstrate direct electrochemistry using the metal organic framework compound. The another method what we have employed to immobilize cholesterol oxidase enzyme is nickel-nickel hydroxide based electrode. We formed nickel-nickel hydroxide electrode on glassy carbon electrode, and it is known nickel-nickel hydroxide electrode is only active in alkaline condition. So we suppress the alkalinity by adding sulfonate molecules so that interfacial acidity is maintained, so that the redox activity of nickel hydroxide is also present, over which cholesterol oxidase enzyme or glucose oxidase enzyme was immobilized, and we can show cholesterol oxidation as well as glucose oxidation on this enzymatic electrode. So these are some of the work carried out with regard to enzymatic oxidation. We have also carried out the reduction of oxygen on bilirubin oxidase. So once this bilirubin oxidase electrode is combined with any of these electrodes, we can form an enzymatic fuel cell. So now coming to the work carried out at SECRI with regard to electrochemical microbial fuel cells. So mostly in the literature, if you see, the key players in the microbial fuel cells are Schwannella bacteria and Geobacter bacteria. Schwannella is a versatile species that is found almost everywhere. It has bioremediation properties. It can metabolize even radioactive uranium. The Geobacter bacteria is known as iron breather. So it lives in environment where there is no oxygen. So Geobacter species and Schwannella is often used in the literature, but at SECRI, we have tried different kinds of microorganisms for direct electron transfer and used these species for current generation in microbial fuel.
It seems ma'am's connectivity is lost. Kindly wait. Kindly be here. She will join anytime. Sheila ma'am, if you can hear us, please try to rejoin the meeting. Leave the meeting and rejoin again.
Dear participants, ma'am will join again. Please wait for a moment.
I'll join. இந்த screen வர மாட்டேங்குது.
Ma'am, just to go to the again the present now again. Go to window and again
Okay.
select the.
Can you hear me?
Yes, ma'am. Yes, ma'am. Only your presentation is not visible. That's all. Ma'am, are you able to open the PPT, ma'am?
I'm trying.
Okay, okay. Ma'am, it is fine. It's please continue, ma'am. Oh.
Is my voice
Yes,
audible?
ma'am. But the presentation is again, yeah, it's opening, ma'am, please.
Now, is it
Yeah, yes,
okay?
ma'am. Yes.
Yes.
Is it okay now?
Yeah,
Yes, ma'am. It's okay now.
yes, please continue, ma'am.
Sorry for the
No, no
interruption.
problem.
Uh, So we have used the different types of microorganisms for the development of microbial fuel cells. We started with Hansanula anemola, an yeast species. We selected this species because it contains the redox enzymes, lactate dehydrogenase, and ferricyanide reductase. So by making use of this organism, we could produce a power output of 2.34 watt per meter cube in presence of carbon felt, and in presence of polyaniline platinum composite, nanocomposite, we could get a power output of 2.9 watt per meter cube. So similarly, hope it's audible. We made use of other species such as Clostridium species and Thiobacillus peroxidans on the cathode side, and we could produce a good amount of power output. And these are the different types of microbial fuel cells that we are using at CSR SICRI, where we have used Acetobacter aceti and Gluconobacter as the biocatalyst. So our paper, our work on Acetobacter and aceti and Gluconobacter, which is published in Environmental Science and Technology, was covered in a different. It was the news was covered in different websites. How bad wine can be used to make a good electrical energy, because these two species, Gluconobacter roseus and Acetobacter aceti, they thrive in wineries. They are responsible for the proliferation of this species in wineries. So we selected this species for the production of electrical energy from bad wine. So this is the coverage given by the ACS journal about our paper, and we could show that we formed three types of microbial fuel cells: Acetobacter alone, Gluconobacter alone, and Acetobacter plus Gluconobacter MFC. So we could obtain a Coulombic efficiency of maximum efficiency of 4.65% and a maximum current density of 4.88 ampere per meter cube and maximum power density of the order of 1.5 watt per meter cube. And these are the different designs of microbial fuel cells that were developed in our lab. And this is another kind of microbial fuel cells that is developed with the help of the microbial species Clostridium acetobutylicum. It is used for treating starch-based effluent. So mostly the microbial fuel cells are being recommended for conversion of waste to energy. So starch-based effluents can be treated with the help of this Clostridium acetobutylicum. The power output of this microbial fuel cell is given here. So we could obtain a good power output of 60 watt per meter cube. And we used two different types of wastewater, biscuit factory wastewater and potato wastewater, potato chips making water. So here 1.4 watt per meter cube of power could be produced by making use of this waste. Also, we try to degrade azo dyes using Hansenula anemola for current generation. So this is a dye-based microbial fuel cell which we have developed in our lab. So where we have used the azo dye methyl red for the generation of electrical energy, and using this 1.416 watt per meter cube could be produced at 11.9 ampere per meter cube, and internal resistance was 357.69. Similarly, in the case of glucose acetate biofuel cell, we could have a higher power output of 3.6 watt per meter cube. And the COD removal in this case was 45.95% within 24 hours, and it reached to 62.16% after 96 hours. This glucose-assisted biofuel cell was gradually increased to 12.72% of COD removal, and it further increased to 40.9% after 96 hours. So this is the example of the microbial fuel cell showing that fuel cells could be applied for running low power density devices like wall clock, digital clock, and it is used for display devices like this. So such devices can be powered by the microbial fuel cells. Then later on, we also try to make use of fuel cells from cellulose degrading bacteria, because glucose is a substance that can be metabolized easily by many of the organisms, but cellulose is a complex substrate which is not easily degradable. So cellulose degrading bacteria, we were on the lookout for cellulose degrading bacteria. So we collected the rumen fluid from the cow or goat, and we used the sample to to isolate the microorganisms present in the rumen fluid that can degrade cellulose. So we were specifically looking for microorganisms that can degrade cellulose. We know the cows and goats, the animals have the capacity to degrade cellulosic composites to a large extent. So we selected the rumen fluid of the cow, and we isolated the cellulose degrading bacteria, and we used it in a microbial fuel cell configuration for producing electrical energy. Then later on, we moved on to the light sensitive microorganisms like oscillatory NA for degrading lignocellulosic compounds. So here we made use of 3-compartment microbial fuel cell. So in the first compartment, hydrolysis of cellulosic compound was happening in presence of light sensitive bacteria, cyanobacteria, and the formed glucose was fed into the our usual microbial fuel cell, which consisted of the catalyst, acetobacter, and gluconobacter. So from the where the electrical energy was produced. So the 3-chamber combination was useful for degrading lignocellulosic biomass. So lignocellulosic biomass polymers was very difficult to degrade under ordinary condition. So we made use of 3 different kinds of bacteria to degrade them and to produce electrical power. And this, in this case, 10 watts per meter cube of power could be produced using this microbial fuel cell. And the same fuel cell, we could extend it by modifying the anode with the chitosan and sodium alginate film, which functioned as the anode. So the cellulose fed 3-compartment MFC produced a maximum power output of 6.62 watt per meter cube at 17.55 ampere per meter cube. Then later on, we studied the bioelectrocatalysis of acetabacter acetate alone by direct electron transfer using template deposited nickel anode. So we made use of carbon paper over which nickel was electro deposited using a template, and over which biofilm was formed, and electrical current generation property of this electrode was studied, and you can see the maximum current that is produced in presence of carbon paper compared with templated carbon paper, nickel carbon paper. You can see from 2.2 to 42.4. The current has increased to a large extent. Similarly, in the case of ethanol, the current has increased from 10.6 to 67.6. Then also we modified the cathode side also. In presence of the anodic film, we modified the cathode side with the Prussian blue so that it enhances oxygen reduction property of the cathode, and we could see how the Coulombic efficiency improved in the presence of the modified cathode. Other type of catalyst we have used are nickel foam modified with polyaniline, titanium carbide, and chitosan as anode material in microbial fuel cells. Here we obtained the maximum power output in this case. So we can see 18 watt per meter cube of power could be obtained in this case in presence of polyaniline titanium carbide catalyst. So coming to the outlook, so these are all the different kind of work that we have carried out at CSR Secre. Coming to the outlook of these biofuel cells, they cannot be used for generation of large amount of power. They can be used for monitoring devices in remote locations, powering electronic devices from renewable energy sources, and further applications of further versions of microbial fuel cells are microbial electrolysis cells and microbial desalination cells. And it can be used as for double purpose for conversion of waste to electricity, also for treating waste as well as to produce electricity. So it is a simultaneous process, and for bioremediation of environmental contaminants. And further, already in literature, there are a lot of applications of microbial fuel cells powering robots. So they can be produced, robots can be produced to collect the garbage in our environment. So that is our vision. So the robots can collect the garbage from the environment, and they can clean the environment as well as produce electrical energy for their own functioning. Also, MFCs producing electricity from urine, providing power for lights, etc., have been already shown. So the synergy, some complementarity between the artificial nanomaterials and biological materials in enhancing electrobiological system is discussed in today's lecture. However, the application is limited by low electron transfer rates within the cells and the cell electrode surface. So therefore, lot of studies are required for long-term operation and scale up of these systems Also, we need to study further regarding stability, viability of these electrode materials, and engineering and bioengineering strategies to boosting efficiency are yet to be studied. Thank you for your kind attention, and thank you for the opportunity given.
Thank you, ma'am. Thank you for your
Thank you.
wonderful lecture. Thank you for the time that you have dedicated to give us this lecture. Now, I request the participants, if you have any doubts or queries, ask them in the chat box. Ma'am will answer these questions. Uh, Ma'am, there is already a question, ma'am. Shall I
Yes.
read it?
Okay.
Yes, ma'am. How much voltage per MFC could be obtained? Is that possible to increase voltage per cell?
And the voltage is decided by the difference in the cell EMF of the anodic and the cathodic reaction. Mostly in our cells, glucose oxidation on the anodic reaction, oxygen reduction on the cathodic reaction. We have obtained around 0.9 volt. The theoretical voltage depends on the thermodynamic EMFs of the anodic reaction and the cathodic reaction.
Thank you, ma'am. There is another question. Is that necessary to supply natural gas such as methane?
No, methane is one of the biofuel that is produced from biological system. It has nothing to do with the biological fuel cell. But nowadays, in the, microbial fuel cells, there is a process called electromethanogenesis. So, where they are trying to produce methane in the anode compartment by additional supply of energy.
Thank you, ma'am. Uh, Participants, if you have any queries, please ask in the chat box. It seems that with the questions.
Okay.
Now, on the behalf of the coordinator, Dr. N. Ponpandian, uh, Professor and Head at Department of Nanoscience and Technology, and the faculty members of Department of Nanoscience and Technology, Bardiyar University, we heartfully thank you, ma'am. Thank you so much for joining us and dedicating your time. You have joined us all the way from US, and it has been a very useful lecture to all of us. Thank you so much for joining, ma'am.
Thank you very much. It's my pleasure to join this team.
Thank you, ma'am. Thank you so much, sir. So much, ma'am.
Thanks.
Yes, ma'am. You may leave the call, ma'am. We will
Okay.
continue. Yes, ma'am. Thank you, ma'am.
Okay.
Yes, ma'am. Participants, please stay in the meeting. Our second speaker will join us very soon. Our our speaker will join very soon, so please stay in the meeting. Good morning, sir. Uh, sir, Ishwarmurthy, sir. Yes, sir.
Yeah.
Welcome, sir. Sir, let me give a
Thank
brief
you.
introduction about you to the participants, sir.
1 minute. Is the course already started or 11:40? Just
Sir,
I
it
want to know the
starts at
timing.
11:45, sir. But if you are free now, we can join. We can start it now itself, sir. We can finish
Okay,
it
that's
earlier,
fine.
or it's according to your wish, sir. Okay, sir.
Okay,
Thank
I
you,
am
sir.
ready. Okay, fine.
So joining us in this session is Dr. Ishwarmurthy Ramasamy, Scientist, Center for Solar Energy Materials, ARCI. Sir has earned his PhD in Energy Engineering from Korean University of Science and Technology. He completed his UG and PG in Physics from Bardar University, Coimbatore. He has over 18 years of expertise in research, working as project associate and visiting fellow and postdoctoral fellowship in various reputed institutions like IIT Madras, Weizmann Institute of Science, Israel, and Pohang University of Science and Technology, Korea. He has been awarded with Dr. APJ Abdul Kalam Gold Medal Award in the year 2015 by GEPRA Delhi. He also has been awarded with uh, Best Brain Korea 21 postdoctoral fellowship in chemical engineering. He is affiliated to various scientific and professional societies, such as he's a member of Material Research Society, and he is a reviewer of various reputed international journals. His field of interest, research interest, includes energy conversion on storage technologies, device physics, optoelectric characterization, and green synthesis of nanomaterials. It's a pleasure and a great opportunity for us to have you, sir. Now you can carry on with your lecture, sir.
Thank you. So I'm trying to share my screen. Still, I'm not getting the option to share.